All right. That's the first kind of big idea what's happening in the chlorophyll. On the side here, we have some water. And water, as you remember, is one of those reactants in photosynthesis. So that water is being used at this point. This is why you need to water plants. And what that water is used for is the splitting of it, or the breaking apart of that water, resupplies electrons to our chlorophyll molecule down here. Because every time that chlorophyll molecule boosts an electron, it's basically like it's leaving a hole behind. It's -1 electron. We don't want to leave chlorophyll without its electrons because that's what has to get boosted over and over again. So the splitting of water releases electrons to refill that chlorophyll molecule. So that's why water is needed for plants to grow. The other thing that is given off in this process by splitting water is oxygen. And remember back in our original reaction again, said that oxygen is a byproduct of photosynthesis. This is the oxygen that's released in the air. Okay, so when we say plants produce oxygen through photosynthesis, this is where the oxygen comes from splitting this water. And then the third thing that's released as a result of splitting water are 2 hydrogen ions. We're not doing anything with those yet, but don't forget about those hydrogen ions because they will come into play. Nothing is wasted here. All right, so let's just review this real quick. What happens in this area? This whole place is called Photosystem 2, or PS2. Again, it's an area in the chloroplast where we've got all sorts of different chlorophyll molecules embedded in the thylakoid membrane. The light comes in, it bounces around between the various chlorophyll molecules. When it eventually hits this superchlorophyll molecule, the P680, that P680 boosts those electrons, or that electron that's in it, so the electron from P680 gets boosted to a higher energy level. In the process, water gets split, which resupplies the original electron that got boosted, produces oxygen gas that goes into the environment, and the hydrogen ions, which we'll get to later. Hopefully that makes sense. We will diagram it again in a little bit. So, like I said, just pay attention for now, and then we'll get to drawing it out in a second. Okay, so we've got to do something with our electron that we just boosted. High energy electrons, if you remember from cellular respiration, high energy electrons are very valuable because they can be used to move things, right? Remember in cellular respiration we had the electron transport chain, and there were these proteins that were embedded in the membrane, and the energy from the electron that was moving from protein to protein was used to transport hydrogen ions to the opposite side of the membrane, and then those hydrogen ions went through the membrane back to where they started to make some ATP. Okay, that was our electron transport chain. We're going to do the same thing in photosynthesis in this particular part of the light reactions. So here we have our proteins embedded in our membrane. Remember, this is the thylakoid membrane that we're imagining. Okay. We use the energy from this high energy electron, which gets passed along. Okay. We use that energy to pump the hydrogen ions to one side of the membrane, and then to let them flow back, and in the process we make some ATP. So again, we have an electron transport chain, the same way we did in cellular respiration, that generates ATP for the cell, using this high energy electron we made in photosystem 1. Or sorry, photosystem 2. Don't get confused about photosystem 2 being first. It's just named photosystem 2 because that was the second photosystem that they discovered. So photosystem 2 is the first one, makes that high energy electron, which gets passed down the electron transport chain to make ATP. Now remember, one of the purposes of the light reactions was to make ATP. So here we go. Check one. Got one of our products made for our light reactions. The other photosystem now that we're getting down to, I'm going to show you here where this label is. It's called photosystem 1. And again, they named this photosystem 1 because this is the one they discovered first. Is another area in the thylakoid membrane with a bunch of chlorophyll. I call it a photosystem because it's kind of all associated together in the same place. The electron that got passed down from the electron transport chain now goes to this chlorophyll molecule right here, P708. Or P700, sorry. Doesn't matter, you don't have to know the names of them. It's just another one of these big supercharged chlorophyll molecules. So that's the end of the electron transport chain. We'll get back to what happens to that electron in a minute because I want to show you over here that this photosystem is also exposed to the light. So photons, which are full of energy that chlorophyll can convert, that energy gets passed from chlorophyll molecule to molecule until it gets to that P700, that supermolecule again. There, the electrons can be charged up again, just like heating up a hot potato, to a high energy level. So that electron that came down the electron transport chain was like a cool potato, right? It lost its energy as it moved from molecule to molecule till it's down to a lower energy level there. But the light that came in could energize it back to up to a higher energy level. So now we've got a high energy electron again, and guess what happens to it? We have another electron transport chain. So that energy can be used to do something. In this case, instead of using the energy to make ATP, we can use the energy to make NADPH. Remember, that's the second molecule that we were making through the light reactions. So we just want to look at this electron transport chain a little bit more closely. This high energy electron is going to be passed along the electron transport chain to activate an enzyme. Just like ATP synthase is what is making ATP down here, NADP reductase is the enzyme that makes NADPH at the end of this electron transport chain. So it's the same idea. We're using electrons to shuttle energy into a chemical reaction. Now, to make NADP into NADPH, all that needs to happen is we need to add on some hydrogens, right? So where do those hydrogens come from? Remember way back here when I said we get back to these hydrogens that came from the splitting of water? Those are the hydrogens that are used to make NADPH over on this side. All right, so NADPH relies on the hydrogens that come from photosystem 2. So now we've made the two main products of the light reactions that I initially discussed. ATP gets made in that electron transport chain between photosystem 2 and photosystem 1. NADPH gets made at the electron transport chain at the end of photosystem 1. Those are the two molecules that we're going to need to go on to the Calvin cycle and make glucose. Remember some of the other products we made in this process though? We also made oxygen. Okay, there's our waste product from photosynthesis giving off oxygen. Okay, as I said, we'll get to the diagrams in a second, but I want to look at one more picture in detail to kind of blow this up into bigger proportion to see how this works in the cell. So we have photosystem 1 and photosystem 2 as they would appear on the thylakoid membrane now. So we'll take a closer look at our diagrams this time. Move that down there. Okay, this whole thing is our chloroplast, and we're looking at one set of granum in the chloroplast, and we're looking at one little thylakoid in that granum. So this whole thing is a thylakoid. Okay, so we're looking at a cross-section of that. It's like if you took a jelly bean and you cut it in half the long way, and now we're looking at it from the side. Okay, that's what we're looking at in this diagram. I want to point out to you, and notice, just because we've talked about this, the thylakoid membrane is a lipid bilayer. Right? You can see the heads and the tails, that the tails are end to end because there's 2 layers of lipids. And then embedded in that lipid bilayer are these photosystems. Okay? Photosystem 2 and photosystem 1. They are a collection of, as I said, of chlorophyll and other proteins embedded in the lipid bilayer. And then we have these proteins, these electron acceptors that are in the electron transport chain, these proteins that are embedded in the membrane. See, here we have a protein that goes all the way through, an integral protein. Here we've got some proteins that don't go all the way through. They're just on the inner layer, so those are peripheral proteins. So it all comes back to things we learned when we were talking about the cell membrane. Down here is another protein that goes through the membrane. In this case, this one's an enzyme. That's that ATP synthase that we talked about when we were doing cellular respiration. Okay, so there's kind of your map of where we are. So let's go through the process one more time, this time in thinking in terms of where we are on the membrane. So the little squiggly yellow line means we got sunlight coming in, exciting our electrons. We have photosystem 2, as I said, I'll put my label back up there, has this chlorophyll that excites the electron up to a higher state. In the process, we split water into oxygen gas, hydrogen ions, and more electrons to resupply that chlorophyll that boosted the original electron. The, where were we here? Electron up here that got boosted to a higher energy level gets passed down the electron transport chain. Okay, in the process, we pump hydrogen ions into this area. Okay, it's called the thylakoid space. And then they will flow out through our ATP synthase to make ATP. In the meantime, that little electron has traveled along and now resupplies photosystem 1 with electron. Light is coming into photosystem 1 as well. It energizes those electrons to a higher energy level. They bounce down a second electron transport chain, which activates an enzyme to make NADPH. Remember, the hydrogens to make NADPH come from photosystem 2. And then both the NADPH and the ATP are going to move on to the Calvin cycle to help to make glucose. All right, hopefully that helps to see you, or helps you to see that in a bigger perspective. All right, the next thing we're going to do is do a diagram for you to get this into your notebook in a way that hopefully makes sense to you.